The 1980s marked the dawn of the personal computer age. It was a time when technology began to infiltrate our daily lives in ways previously unimaginable. This revolution transformed how we live, work, and interact with the world. Personal computers became a staple in homes and offices, changing the landscape of communication and productivity. Today, we stand at the cusp of another revolution, one with the potential to be even more transformative: genetic engineering. This new frontier holds promises that could redefine the very essence of life itself. This field, dedicated to manipulating the very building blocks of life, promises to reshape our world in ways we can only begin to imagine. From curing genetic diseases to enhancing human capabilities, the possibilities are endless. The journey of genetic engineering began long before we understood the intricacies of DNA. Early scientists and researchers laid the groundwork for what would become a revolutionary field. For millennia, humans have been shaping the genetic makeup of plants and animals through selective breeding. This ancient practice was our first foray into genetic manipulation. By choosing which organisms reproduced, our ancestors enhanced desirable traits over generations, creating stronger, more resilient species. From the size of their crops, ensuring bountiful harvests to sustain growing populations, to the temperament of their livestock, making them easier to manage and more productive. This ancient practice, while effective, was a shot in the dark, relying on trial and error rather than a deep understanding of the underlying mechanisms. Today, with our advanced knowledge of genetics, we can approach this age-old practice with precision and purpose. The discovery of DNA in the mid-20th century was a watershed moment. This elegant double helix carrying the code of life unlocked the secrets of heredity and opened up a new realm of possibilities. Scientists could finally understand the blueprint of life, paving the way for deliberate and targeted genetic modifications. The 1960s and 70s saw the first forays into this new frontier. Early techniques were rudimentary, often involving exposing organisms to radiation to induce random mutations. This process, while capable of producing desirable traits, was uncontrolled and unpredictable. However, these early experiments, however crude, marked the first steps towards manipulating the code of life itself. Scientists also began experimenting with inserting genetic snippets into bacteria and animals, laying the groundwork for future breakthroughs. The birth of the first genetically modified animal in 1974, a humble mouse, marked a pivotal moment. This breakthrough demonstrated the possibility of altering the germline of an organism, meaning the changes would be passed down to future generations. This development opened up new avenues for research, allowing scientists to study the role of specific genes in health and disease. The 1980s saw the first patent for a genetically modified microbe, a bacterium engineered to consume oil spills. This event marked the beginning of the commercialization of genetic engineering, highlighting its potential in addressing environmental challenges. The decade culminated with the Flavor Savr tomato in 1994, the first genetically modified food available to consumers. Engineered to have a longer shelf life, the Flavor Savr tomato represented the potential of genetic engineering to revolutionize agriculture and food production. Despite these advancements, early genetic engineering techniques were often costly, time-consuming, and lacked precision. The true game changer arrived with the advent of CRISPR technology in the early 21st century. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing tool that has democratized access to genetic engineering. CRISPR's origins lie in the defense mechanisms of bacteria. These single-celled organisms use CRISPR systems to target and destroy invading viruses by cutting their DNA. Scientists quickly realized the potential of this system for precise gene editing. By adapting the bacterial CRISPR system, researchers can now target and edit specific DNA sequences with unprecedented accuracy and efficiency. The CRISPR technology has dramatically reduced the cost and time required for gene editing, making it accessible to a wider range of researchers and accelerating the pace of discovery. Section 5. CRISPR in action, healing the future. The applications of CRISPR technology are vast and continue to expand at an astonishing rate. From agriculture to medicine, this revolutionary tool is reshaping our approach to some of the most challenging problems. In medicine, CRISPR holds immense promise for treating a wide range of diseases. Its precision allows for targeted interventions that were previously unimaginable. Scientists have already demonstrated its potential in treating HIV by successfully cutting the virus out of human cells. This groundbreaking work is paving the way for new treatments. This breakthrough offers hope for a cure for HIV and other retroviruses, such as herpes, which have long eluded effective treatments. The implications for global health are profound. CRISPR also has the potential to revolutionize cancer treatment. By enabling precise targeting of cancer cells, it could lead to therapies that are both more effective and less toxic. By enhancing the ability to target and eliminate cancer cells, CRISPR could lead to more effective and less toxic cancer therapies. This could significantly improve patient outcomes and quality of life. The technology also offers hope for treating genetic diseases. Many of these conditions are currently incurable and can be devastating for families. Many of these debilitating and often fatal conditions are caused by single-letter errors in our DNA. Correcting these errors at the genetic level could transform lives. CRISPR provides the tools to correct these errors, potentially eradicating thousands of genetic diseases. The future of medicine is being rewritten one gene at a time. Section 6. Germline Editing, A Brave New World. One of the most ethically charged applications of CRISPR is germline editing. This involves making genetic modifications to reproductive cells or embryos, meaning the changes would be passed down to future generations. Germline editing has the potential to eliminate inherited diseases, but it also raises profound ethical concerns. Critics argue that germline editing could lead to the creation of designer babies, where parents could choose their children's traits, from eye color to intelligence. This raises concerns about exacerbating social inequalities and undermining the very concept of human diversity. Furthermore, the long-term consequences of altering the human gene pool remain unknown, raising concerns about unintended consequences for future generations. Section 7. The Scales of Acceptance. Society and Genetic Engineering. As genetic engineering becomes increasingly powerful and ubiquitous, societal acceptance will play a crucial role in shaping its development and application. The initial public reaction to genetically modified organisms, or GMOs, was mixed, with concerns about potential environmental and health risks. However, as GMOs have become more prevalent in our food supply, public acceptance has gradually increased. This trend suggests that as genetic engineering technologies become more familiar and their benefits become more apparent, public acceptance will likely continue to grow. Open and transparent communication about the risks and benefits of genetic engineering will be essential in fostering public trust and ensuring its responsible development. Section 8. A Future Written in Code: Possibilities and Risks. The potential applications of genetic engineering extend far beyond medicine and agriculture. This revolutionary technology holds the promise of transforming various sectors, from environmental conservation to industrial manufacturing. Scientists envision a future where genetic engineering could help us address some of the world's most pressing challenges. Imagine a world where diseases are eradicated and food scarcity is a thing of the past. For example, genetically engineered microbes could be used to clean up pollution, produce biofuels, and even help in waste management by breaking down plastics and other non-biodegradable materials, or even manufacture new materials. These innovations could lead to more sustainable and efficient production processes, reducing our reliance on finite natural resources. CRISPR could potentially be used to slow down or even reverse the aging process, offering the possibility of extending human lifespan and improving the quality of life in our later years, raising profound questions about overpopulation and resource management. How will societies adapt to a world where people live significantly longer lives? However, alongside these exciting possibilities loom potential risks. The same technology that holds so much promise also carries significant ethical and safety concerns. One concern is the possibility of off-target edits, where CRISPR inadvertently modifies genes other than the intended target. This could lead to unintended consequences that we are not yet fully prepared to handle, where CRISPR inadvertently modifies genes other than the intended target. These unintended edits could have unforeseen and potentially harmful consequences, affecting not just individuals but entire ecosystems. These unintended edits could have unforeseen and potentially harmful consequences. The ripple effects of such changes could be far-reaching and difficult to predict. Another concern is the potential for genetic engineering to be used for malicious purposes. The same tools that can heal could also be weaponized. For example, the technology could be used to create genetically enhanced soldiers, raising ethical questions about the future of warfare and human rights, or to develop new biological weapons. The potential for misuse adds a layer of complexity to the already intricate ethical landscape of genetic engineering. Section 9. Navigating the genetic frontier. A call for responsibility. Genetic engineering stands as one of the most powerful tools humanity has ever wielded. Its potential to improve human health, address global challenges, and reshape our world is immense. However, this power comes with a profound responsibility. As we venture further into the genetic frontier, we must proceed with caution, carefully weighing the potential benefits against the risks. Open and transparent dialogue, rigorous ethical frameworks, and stringent safety regulations will be essential to ensure that genetic engineering is used responsibly and for the benefit of all humankind. The future is written in code, and it is up to us to ensure that it is a future worthy of our aspirations.